Welcome to the fascinating world of proteomics data analysis. In this video, we'll explore how scientists study proteins on a large scale, using advanced technologies and computational tools. Proteomics is the large-scale study of proteins, which are essential molecules that perform a vast array of functions within living organisms. Understanding proteins and their interactions helps researchers uncover the mechanisms behind health and disease. As we journey through this topic, we'll break down complex concepts and provide practical examples, making proteomics data analysis approachable for everyone, whether you're a student, researcher, or simply curious about the science of life. Proteomics refers to the comprehensive study of the entire set of proteins produced or modified by an organism. Unlike genomics, which focuses on genes, proteomics looks at the dynamic protein landscape, including their structures, functions, and interactions. This field provides insights into how proteins work together to maintain cellular processes and respond to changes in the environment. By analyzing proteomes, scientists can identify biomarkers for diseases, discover new drug targets, and better understand biological pathways. Let's dive deeper into the core concepts that make proteomics such a powerful tool in modern biology. Proteomics plays a crucial role in advancing our understanding of biology and medicine. By studying the full range of proteins in a cell, tissue, or organism, researchers can uncover how diseases develop and progress. Proteomics data analysis helps identify specific proteins associated with certain conditions, paving the way for early diagnosis and personalized treatments. Additionally, it aids in drug discovery by revealing potential targets for new therapy. The ability to analyze vast amounts of protein data has transformed biomedical research, making proteomics an indispensable part of life sciences today. While both proteomics and genomics are essential fields in molecular biology, they focus on different aspects. Genomics studies the complete set of genes, providing the blueprint for life. Proteomics, on the other hand, examines the proteins that are actually expressed, modified, and functional within cells. This distinction is important because proteins are the molecules that carry out most cellular activities. By comparing proteomics and genomics, researchers gain a more comprehensive view of biological systems, bridging the gap between genetic information and functional outcomes. Typical proteomics workflow involves several key steps, starting with sample preparation. Proteins are extracted from biological samples, such as cells or tissues, and then processed for analysis. Next, proteins are separated and identified using techniques like mass spectrometry. The resulting data is then analyzed using computational tools to interpret protein identities, quantities, and modification. Each step in the workflow is critical for ensuring accurate and meaningful results. By following a systematic approach, researchers can uncover valuable insights into protein function and regulation. Sample preparation is the foundation of any proteomics experiment. This step involves collecting biological material, such as blood, tissue, or cultured cells, and extracting proteins from these samples. Proper sample handling is essential to prevent protein degradation and contamination. Researchers often use buffers and detergents to solubilize proteins, followed by techniques like centrifugation to remove unwanted debris. The quality of sample preparation directly impacts the reliability of downstream analysis, making it a crucial part of the proteomics workflow. Extracting proteins from samples requires specialized methods to ensure maximum yield and purity. Common techniques include mechanical disruption, such as sonication or homogenization, and chemical lysis using detergents or enzymes. The choice of extraction method depends on the sample type and the proteins of interest. Efficient extraction is vital for obtaining a representative snapshot of the proteome, which is necessary for accurate downstream analysis. Researchers must carefully optimize protocols to balance protein yield, integrity, and compatibility with analytical techniques. After extraction, quantifying the amount of protein in each sample is essential. Accurate quantification ensures that equal amounts of protein are analyzed across samples, which is critical for reliable comparisons. Common quantification methods include the Bradford assay, BCA assay, and Lowry assay, each with its own advantages and limitations. Proper quantification helps standardize experiments and improves the reproducibility of results. By carefully measuring protein concentrations, researchers set the stage for successful proteomics analysis. Proteins in complex samples must be separated before identification. Techniques such as gel electrophoresis and liquid chromatography are commonly used for this purpose. Gel electrophoresis separates proteins based on size and charge, while chromatography separates them based on chemical properties. These methods help reduce sample complexity, making it easier to identify individual proteins. Effective separation is a key step in the proteomics workflow, enabling high-resolution analysis and accurate protein identification. Mass spectrometry is a powerful analytical technique used to identify and quantify proteins in proteomic studies. It works by ionizing protein fragments and measuring their mass-to-charge ratios. The resulting spectra provide detailed information about protein identities and modifications. Mass spectrometry offers high sensitivity and accuracy, making it the gold standard for proteomics analysis. Understanding the basics of this technology is essential for interpreting proteomics data and drawing meaningful conclusions from experiments. There are several types of mass spectrometers used in proteomics, each with unique features. Common types include time-of-flight TOF, quadrupole, and Orbitrap instruments. Each type offers different advantages in terms of resolution, sensitivity, and speed. The choice of instrument depends on the specific requirements of the experiment, such as the complexity of the sample and the desired level of detail. By selecting the appropriate mass spectrometer, researchers can optimize their proteomics workflows for the best results. Once proteins are separated and analyzed by mass spectrometry, the next step is identification. This involves matching the observed spectra to known protein sequences in databases. Sophisticated algorithms compare the mass and fragmentation patterns of peptides to reference data, allowing researchers to determine which proteins are present in the sample. Accurate identification is crucial for understanding biological processes and drawing meaningful conclusions from proteomics experiments. In addition to identifying proteins, quantifying their abundance is a key goal in proteomics. There are two main approaches: label-based and label-free quantification. Label-based methods use chemical or isotopic tags to compare protein levels across samples, while label-free methods rely on the intensity of mass spectrometry signals. Each approach has its strengths and limitations, and the choice depends on the experimental design and available resources. Accurate quantification provides insights into protein expression changes under different conditions. Proteomics data can be acquired using different strategies, such as data-dependent acquisition (DDA) and data-independent acquisition (DIA). DDA selects the most abundant ions for analysis, while DIA collects data on all ions within a specific range. Each strategy offers unique advantages in terms of coverage, sensitivity, and reproducibility. Choosing the right data acquisition method is essential for obtaining high-quality proteomics data that meets the goals of the study. After data acquisition, raw mass spectrometry data must be processed before analysis. This involves converting instrument files into standardized formats, filtering noise, and extracting relevant features. Specialized software tools automate these steps, ensuring consistency and accuracy. Proper data processing is vital for downstream analysis, as it affects the quality and reliability of the result. By following best practices, researchers can maximize the value of their proteomics data. Database searching is a critical step in proteomics data analysis. It involves comparing experimental spectra to theoretical spectra generated from protein databases. Popular search engines like Mascot, Sequest, and MaxQuant are used to identify peptides and proteins. The accuracy of database searching depends on the quality of the database and the search parameters. By optimizing these factors, researchers can improve identification rates and reduce false positives. Controlling the false discovery rate, FDR, is essential for ensuring the reliability of proteomics results. FDR measures the proportion of incorrect identifications among all positive results. Researchers use statistical methods such as target decoy approaches to estimate and control FDR. Setting appropriate thresholds helps balance sensitivity and specificity, reducing the risk of reporting false positives. Effective FDR control is a hallmark of high-quality proteomics data analysis. Validating identified peptides is a crucial step in proteomics analysis. This process involves confirming that the detected peptides are truly present in the sample, and not artifacts of the analysis. Researchers use criteria such as spectral quality, retention time, and reproducibility to validate peptides. High-confidence peptide validation increases the reliability of protein identification and ensures that downstream analyses are based on accurate data. Protein inference is the process of determining which proteins are present in a sample based on the identified peptides. Since some peptides can be shared among multiple proteins, this step requires careful analysis to avoid ambiguity. Advanced algorithms and statistical models help resolve these challenges, providing a more accurate picture of the proteome. Proper protein inference is essential for drawing meaningful biological conclusions from proteomics data. Quantitative analysis in proteomics involves comparing protein abundances across different samples or conditions. Statistical methods are used to identify significant changes in protein expression, which can reveal important biological insights. Tools like normalization and scaling help account for technical variability, ensuring that observed differences are biologically meaningful. By applying robust quantitative analysis, researchers can uncover patterns and trends that drive scientific discovery. Statistical analysis is a cornerstone of proteomics data interpretation. Researchers use a variety of statistical tests, such as t-tests and ANOVA, to determine whether observed differences in protein levels are significant. Multiple testing correction methods like the Benjamini-Hochberg procedure help control for false positives. Proper statistical analysis ensures that conclusions drawn from proteomics data are robust and reliable, supporting sound scientific finding. Bioinformatics tools play a vital role in proteomics data analysis. These software applications help process, visualize, and interpret complex datasets. Popular tools include MaxQuant, Proteome Discoverer, and Skyline, each offering unique features for different analysis needs. By leveraging bioinformatics, researchers can handle large-scale data efficiently and extract meaningful biological information. Staying updated with the latest tools and techniques is essential for successful proteomics research. Visualizing proteomics data helps researchers identify patterns, trends, and outliers. Common visualization techniques include heat maps, volcano plots, and principal component analysis (PCA) plots. Effective visualization makes complex data more accessible and supports clear communication of results. By presenting data visually, scientists can share their findings with broader audiences and facilitate collaboration across disciplines. After identifying and quantifying proteins, researchers often perform functional annotation to understand their roles in biological processes. This involves mapping proteins to known pathways, molecular functions, and cellular components using databases like gene ontology (GO) and KEGG. Functional annotation provides context for the data, helping researchers interpret the biological significance of their findings and generate new hypotheses for further study. Pathway analysis is a powerful approach for understanding how proteins interact within biological systems. By mapping identified proteins to known pathways, researchers can uncover how cellular processes are regulated and how they change in response to different conditions. Pathway analysis tools, such as Ingenuity Pathway Analysis and Reactome, help visualize these interactions and identify key regulatory nodes. This step bridges the gap between raw data and biological interpretation. Proteins rarely act alone. They interact with other proteins to carry out complex functions. Protein-protein interaction (PPI) networks visualize these relationships, revealing clusters and hubs that play central roles in cellular processes. Tools like string and cytoscape are commonly used to construct and analyze PPI networks. Understanding these networks helps researchers identify potential drug targets and gain insights into disease mechanism. Proteins often undergo post-translational modifications, PTMs, such as phosphorylation, glycosylation, and ubiquitination. These modifications regulate protein function, stability, and interactions. Detecting and analyzing PTMs is a key aspect of proteomics, as they can have significant biological effects. Advanced mass spectrometry techniques and specialized software are used to identify and quantify PTMs, providing deeper insights into protein regulation. Ensuring data quality is essential for reliable proteomics analysis. Quality control (QC) measures include monitoring instrument performance, checking sample integrity, and validating results with standards. Regular QC helps detect issues early, preventing costly errors and ensuring reproducibility. By implementing robust QC protocols, researchers can trust their data and draw confident conclusions from their experiment. Data normalization is a crucial step in proteomics analysis, ensuring that technical variations do not obscure true biological differences. Normalization methods adjust for factors like sample loading, instrument drift, and batch effects. Common approaches include total ion current normalization and median normalization. Proper normalization improves the accuracy and comparability of results, supporting robust scientific conclusion. Batch effects are unwanted variations that arise from differences in sample processing, instrument runs, or experimental conditions. Correcting for batch effects is essential for accurate proteomics analysis. Statistical methods such as combat and empirical Bayes approaches are used to adjust data and minimize these variations. Proper batch effect correction ensures that observed differences reflect true biological changes, not technical artifacts. Missing data is a common challenge in proteomics, often resulting from low abundance proteins or technical limitations. Handling missing data appropriately is important for accurate analysis. Strategies include imputation, where missing values are estimated based on observed data, and filtering, where incomplete data points are removed. The choice of method depends on the extent and pattern of missingness. Proper handling ensures that results are not biased or misleading. Reproducibility is a cornerstone of scientific research, and proteomics is no exception. Ensuring that results can be replicated by independent researchers builds confidence in findings. Key practices include using standardized protocols, sharing raw data, and documenting analysis workflows. By prioritizing reproducibility, the proteomics community advances knowledge and fosters collaboration. Sharing proteomics data promotes transparency and accelerates scientific progress. Public repositories like PRIDE, Peptide Atlas, and MassIVE store raw and processed data, making it accessible to the global research community. Data sharing enables reanalysis, validation, and meta-analysis, driving new discoveries and supporting open science initiatives. Integrative omics combines proteomics with other omics data, such as genomics, transcriptomics, and metabolomics. This holistic approach provides a more comprehensive understanding of biological systems. By integrating multiple data types, researchers can uncover complex relationships and gain deeper insights into health and disease. Integrative omics is at the forefront of systems biology and personalized medicine. Proteomics has significant applications in clinical research and medicine. By analyzing patient samples, researchers can identify disease biomarkers, monitor treatment responses, and develop personalized therapies. Clinical proteomics is transforming diagnostics and paving the way for precision medicine, where treatments are tailored to individual patients based on their unique protein profiles. Drug discovery benefits greatly from proteomics data analysis. By identifying proteins involved in disease pathways, researchers can discover new drug targets and evaluate the effects of potential compounds. Proteomics also helps assess drug safety and efficacy, supporting the development of more effective and safer medications. This integration accelerates the drug discovery process and improves patient outcome. Despite its power, proteomics data analysis faces several challenges. These include sample complexity, data volume, technical variability, and missing values. Addressing these challenges requires careful experimental design, advanced analytical methods, and continuous technological innovation. By overcoming obstacles, researchers can unlock the full potential of proteomics for scientific discovery. Proteomics is a rapidly evolving field with new technologies constantly emerging. Advances in mass spectrometry, sample preparation, and data analysis are expanding the capabilities of proteomics research. Techniques like single-cell proteomics and data-independent acquisition are pushing the boundaries of what's possible, enabling deeper and more precise analysis of the proteome. Future of proteomics data analysis is bright, with ongoing innovations promising even greater insights. Integration with artificial intelligence and machine learning is expected to revolutionize data interpretation, making analyses faster and more accurate. As technology advances, proteomics will continue to play a central role in understanding biology and improving human health. As with any powerful technology, ethical considerations are important in proteomics research. Issues such as data privacy, informed consent, and responsible data sharing must be addressed. Researchers should follow ethical guidelines and ensure that their work benefits society while respecting individual rights. By maintaining high ethical standards, the proteomics community builds public trust and supports responsible innovation. Proteomics community is supported by a wealth of resources, including online forums, conferences, and training programs. These resources foster collaboration, knowledge sharing, and professional development. Engaging with the community helps researchers stay informed about the latest advances and best practices in proteomics data analysis. Let's look at a real-world example of proteomics in action. In disease biomarker discovery, researchers analyze patient samples to identify proteins that are uniquely associated with a specific condition. By comparing protein profiles between healthy and diseased individuals, scientists can pinpoint potential biomarkers for early diagnosis and targeted treatment. This case study highlights the practical impact of proteomics data analysis in improving healthcare outcomes. If you're new to proteomics data analysis, start by learning the basics of protein biology and analytical techniques. Familiarize yourself with common software tools and best practices for data processing. Don't hesitate to seek guidance from experienced researchers or join online communities. With patience and practice, you'll build the skills needed to tackle complex proteomics datasets and contribute to exciting discoveries. Let's recap what we've covered in this video. We explored the fundamentals of proteomics, the workflow from sample preparation to data analysis, and the importance of quality control and reproducibility. We discussed key analytical techniques, bioinformatics tools, and real-world applications in medicine and drug discovery. By understanding these concepts, you're well equipped to appreciate the power and potential of proteomics data analysis. As proteomics continues to evolve, staying informed and adaptable is key. Embrace new technologies, collaborate with others, and keep learning. The field offers endless opportunities for discovery and innovation. Whether you're a student, researcher, or enthusiast, your journey in proteomics data analysis is just beginning. Thank you for joining us, and we wish you success in your scientific endeavors.